When it comes to predicting outcomes with CAR T-cell therapy treatment, looking at patient and disease characteristics, as well as the CAR T-cell product attributes, really will influence both the efficacy and safety endpoints of nearly all clinical cell therapy trials. So trying to quantify all of these different contributions remains a particular challenge, and the purpose of this particular study was to try to improve the understanding of each of these attributes and their influence on CAR T-cell therapy outcomes. So the authors performed a systematic examination of patient features, T-cell starting material attributes, and then the CAR T-cell drug product attributes using a random forest-based multifactorial analysis. And the data they obtained was from 172 patients who had undergone CAR T-cell therapy with lisocabtagene on the Transcend NHL 001 clinical trials. The endpoints that they looked at were the impact on the pharmacokinetics, efficacy endpoints, generally CR and survival outcomes, and then the safety endpoints, specifically looking at CRS and neurotoxicity. A total of 238 features were studied, but 136 were relating to the cell product itself, and there were 102 patient characteristics included. And then this was further analyzed as 76 independent features and analyzed as independent clusters. So 33 independent factors for cell product and 47 patient characteristics. So when you're looking at the various endpoints, starting with the pharmacokinetic features, the younger the age of the patient and those with fewer lines of treatment resulted in higher CAR T-cell expansion, and also the composition of the product was more naive in that particular, in those particular populations. So having a less effector cytokine secretion, i.e. more naive CD8 positive T-cell drug product was also associated with higher CAR T-cell expansion. When looking at efficacy endpoints, things like having a higher functional cytokine production in the antigen-specific CD4 positive cells in the lisocabtagene drug product itself was associated with a longer PFS. Patients with reduced tumor burden was associated with a longer PFS. And then patients who had the indication of either transformed follicular lymphoma or primary mediastinal B-cell lymphoma was also associated with a longer PFS. There's something about these subtypes that make them more responsive to this treatment. CR rates were influenced by higher antigen-specific functional cytokine production, both in the CD4 and CD8 components of the lisocabtagene product. And again, the lower tumor burden in patients was associated with improved CR rates. When looking at the toxicity, specifically the CRS and any grade neurotoxicity, greater tumor burden, and patients who required bridging therapy were more likely to have higher rates of any grade CRS or neurotoxicity. So taken together, the major findings from this study really reinforce what we already know based on other studies, and that's really that the tumor burden and that the antigen-specific function of the drug product are the most important factors that are associated with the CAR T-cell expansion, safety of the product, as well as efficacy of the product. So the idea was to identify areas or potential opportunities for either combination treatments or manufacturing strategies to try to manipulate the cell product and ideally improve the clinical response. So although this is not ready to be translated to clinical practice, it does give the next kind of research questions moving forward to try to improve the outcome of this one-time treatment for these patients.